Hi, my name is Knox, and I'm going to give you an audio tour of this article. Single prolonged stress refers to serial exposure to restrained phosphorus and ether, and this stress protocol induces a number of symptoms that mimic PTSD symptoms. These include enhanced arousal, enhanced negative feedback of the HPA axis, and enhanced glucocorticoid receptor expression. You can pair single prolonged stress with Pavlovian fear conditioning, and typically SPS animals don't show changes in acquisition or expression of fear memory or acquisition of extinction memory, but they show enhanced freezing during an extinction test. So they have an extinction retention deficit. So in a previous study, we had shown that SPS enhanced hippocampal GR expression and SPS induced extinction retention deficits might be linked, but we didn't really know how. So one thing that we were thinking is that if you have enhanced hippocampal GR expression, during fear conditioning, you might have enhanced GR activation because of enhanced cord synthesis. And so in SPS rats, you might have enhanced GR cord binding. During fear conditioning, this would strengthen fear memory, which could be the cause of extinction retention deficits in the SPS model. So to examine this somewhat, we inhibited cord synthesis prior to fear conditioning and hypothesized that this treatment would attenuate extinction retention deficits in SPS rats. So this is the methods that we used. We did single prolonged stress or control procedure, 90 minutes before administered meteropone, then did fear conditioning, extinction training, and extinction testing. So here are the results. SPS enhanced acquisition of fear conditioning. So the SPS animals together had higher levels of freezing, particularly on trials 3, 4, and 5, and a little bit 2. But there were no drug effects and no stress by drug interaction. SPS also disrupted acquisition of fear extinction. So at the end of the extinction training session, there was enhanced freezing in the SPS rats as a group. But again, there were no drug or stress by drug interactions, which suggests administering meteropone prior to fear conditioning had no effects on expression of fear or extinction. So there are some interesting findings here. SPS enhanced baseline freezing prior to CS presentations during the extinction tests. This was not observed during extinction training, which suggests SPS animals developed contextual fear to a context that was never actually paired with a foot shock, so second order contextual fear conditioning. SPS animals also showed enhanced freezing to the CS presentations, which is your standard extinction retention deficit. And SPS animals that received 25 mg per kg of meteropone prior to fear conditioning had the greatest enhancement in freezing. So inhibiting cord synthesis during fear conditioning actually made things worse. It didn't attenuate extinction retention deficits in SPS rats. Interestingly, meteropone administration prior to fear conditioning in control rats had no effect on extinction memory. So the results suggest that inhibiting cord synthesis during fear memory formation exacerbates extinction retention deficits in SPS rats. In turn, this suggests that potentially GR cord binding during fear memory formation prevents extinction retention deficits in the SPS model from being as bad as it otherwise would be. So you could hypothesize based on these results that SPS-induced changes in GR function may be adaptive, as it potentially facilitates extinction retention either by inhibiting fear memory or somehow changing neural circuits that facilitate acquisition of extinction. Of course, the results for this study only suggest that SPS-induced changes in GR function can be adaptive. In future, we want to actually demonstrate this, and that would involve directly manipulating GRs to show that activation of GRs in specific brain regions during fear conditioning attenuates extinction retention deficits in the SPS model, while antagonizing or blocking GR receptors in these same brain regions kind of mimics the effect that we saw in this study and enhances or exacerbates extinction retention deficits in SPS rats. I'd like to acknowledge everyone at the University of Michigan, where I was a postdoc, who helped me with this research, and everyone in my lab, in particular Jordan and Kevin, who were undergrads that really helped with a lot of the behavioral stuff. Thanks so much for your time.